My name is Dion Krupa, and I am the program manager with the Innovate Bio grant at Austin Community College in Austin, Texas, and I am your host today. Before we begin, I have a few housekeeping announcements. Please feel free to use the chat box for any questions that you may have for any of our speakers today. And then additional information on the project talks can be found on the innovatebio.org website that I will drop in the chat. And then lastly, we respectfully ask that you complete a survey at the end of this talk so that it will help us in planning future events. So it is with great pleasure that I announce our Project Talk speakers. Our main speaker is Dr. Sandra Porter. She will be joined by Drs. Todd Smith and Margaret Bryans. The title of their presentation is Using Hackathons to Catalyze Research Projects in Antibody Engineering. Please join me in welcoming Sandy, Todd, and Maggie.
Thanks, Dion. Okay, it's great to see all of you. And I guess without further ado, I will get started. All right. Okay, so our project, our AT project is titled Developing Cures, Course-Based Undergraduate Research Experiences for Antibody Engineering. This is a little bit out of order. I'm going to talk first and describe what hackathons are all about and our approach to hackathons. And then Todd is going to talk, and then Maggie, who has a lot to say about NISTCHO cells, will talk about the NISTCHO cell part of the project. Okay, there we go. All right, so yeah, I just said that we have links to some of these things. I will be putting links to all of our resources on the presentation page when the presentations get posted, so no need to worry about that. And I want to tell you too that Innovate Bio is going to be hosting a virtual hackathon. This will be next June, the 22nd through 24th. And so this is part of why I want to give you an idea about what hackathons are all about, because you'll have an opportunity to work with your Innovate Bio colleagues on fleshing out your favorite ideas and things that you could do to support biotechnology education. So before we go any further, what is a hackathon? Because I know a lot of people are sometimes puzzled about this. And it is an event where we have teams. The teams work together to create products. And this is a short amount of time. Surprisingly short, but it's surprisingly occupied. The products that we have been working to make through our hackathons are research projects, protocols for the research projects, data sets, any kind of materials, explained materials, dictionaries, anything related to antibody engineering. And the projects are being used in course-based research classes, and they're being used as independent student research projects as well. And we've had a lot of publications I'll show you a little bit later. But hackathons don't just develop research projects, and our goal was never just to develop research projects. I do a lot of coaching for students on what they might need, what skills they might need to bring with them when they're looking for jobs in the workforce. And what they can do through the hackathons is they also develop community. They increase their social capital. I've noticed a lot of our students now are connected to people all throughout the country on LinkedIn. These are connections they never would have made if they hadn't been working on, they hadn't met each other through the hackathons. They get an opportunity to demonstrate that they can contribute to a project, that they're reliable, that they are willing to do things, that they are willing to work as a team, willing to help each other. And this kind of goes along with this idea that you dig your well before you're thirsty, because those networks are going to be very important to them when it comes time to look for a job and get references and find jobs. More people get hired because of the people they know working at a company than just through cold calls or resumes. They get hired because there are people they know that already work there that can vouch for them being a reliable person and a hard worker. So a hackathon gives them an opportunity to demonstrate that. Okay, so what does this mean for us? I know most of you have participated in faculty development workshops, trainings, things of that sort. But a hackathon is really different from that, and it's a completely different paradigm. It's really science as a collaborative process. So like if you went to a DNA barcoding workshop, you would hear about DNA barcoding, you'd get some opportunity to be in the lab. But if you were in a hackathon, you'd be developing how to do that, how to use that barcoding to do a research project and develop the whole research project around it. Now, these can be really intense. You're working in teams, you're presenting. They really facilitate deep learning. People can get a really good understanding of something quickly. You can work with a broad group of people, which we'll talk about in a couple of minutes. And through successive hackathons, you can build on previous work. As Maggie will tell you, this can be really productive. And I already mentioned you can be building community. So a difference, again, also between a hackathon and a workshop is that you're in a team, and in each team, you have an assigned role. This is to make your work more efficient because you have a short amount of time. So there's a team leader, there is a writer who's recording what goes on, there are people who go hunt down data, there might be people who are doing coding, might be people who are doing the slides for the presentations. Artists, for example, might be making logos. We have subject matter experts, perhaps, who explain the research papers to other people in the group. Presenters, tech support, who help people with the software. We might have artists. So we can have people fulfilling a lot of different roles. And this helps people work together without tripping over each other because we don't have people competing with each other trying to all do the same thing. So some of the, we have found people sometimes have misconceptions about hackathons and what this means. They do not have to require coding. In fact, ours do not. They don't have to be a competition. That is, teams don't need to be competing with each other. We can all work together and help each other learn. We do not need everybody to have deep expertise or to do lab work or to travel. We've been able to do hackathons with Zoom, which I know some of you may have felt like you've had enough Zoom, but it can still be productive and you can still get a lot out of it. They do require, though, a time commitment, and they do require communication and participation. And at least in terms of the team leaders, it's helpful if you have some preparation. They do require teamwork, and we do have to organize things. I want to tell you a little bit about how this works and what our schedules are like. This was the schedule from last summer. So before the hackathon, we had all of our announcements to recruit participants. They submitted applications. We developed a mailing list. We went through the applications. We sent acceptance letters to the participants. We had them confirm that they were going to attend or not if things come up, right? We assigned them to teams based on their interests and what they asked to do. We set up Slack accounts for everyone so we could get them communicating with each other, finalize their schedule, and we had a pre-hackathon orientation to tell them how the hackathon was going to work and what was going to be involved. The first day, we had an introduction, and then teams started working together. They picked their roles, they decided on an approach to a problem, how they were going to approach it in the workflow, and then we had, every day we had a meeting of the team leaders at the end of the day. The second day and third day, we had guest speakers. On the second day, each team presented what they were going to do, what their projects were going to involve. We had all of our writers met to learn what their responsibilities were going to be. And then I said the team leaders meet. So So each day there were presentations. The first day, as I mentioned, the teams present what they're going to do. The next day, teams present any kinds of blockers or problems, maybe a little bit of a progress report. And the last day, the teams gave final presentations to talk about what they had accomplished and where they might go from there. One thing that we did a lot, since each team might be 4 to 10 people, we tried to have as many people present as possible. And wherever possible, we tried to get students to present if we had student members on the team. So who participated in this? In our last hackathon 2024, we had 8 teams. The team leaders were from 7 different states. 4 of them were new to the project. And then we had some other people who were I would have called co-leaders who were helping. We had, I said 51 people. Of these people, 18 were people that were returning from previous hackathons. In fact, some of them were students who were returning, who had participated in previous hackathons. So last year, about almost half were students. We had a good number of faculty, and we found that there are people from industry and government institutions or research institutions who like to participate also. Sometimes as team leaders, sometimes as in order to learn new skills and develop new techniques and to build their networks. Our participants, about half were from community college, but they were we had a good number from industry and government agencies like the NCBI or different companies. We had some high school students. We were trying not to, but we did anyway. University students. We had people from four-year colleges and state colleges as well. And through this, it has been surprisingly productive. We have 3 publications in cubes that people can use for planning using their own undergraduate projects. We've had 9 publications in peer-reviewed journals, and 5 of those students wrote them. And you can see I have a long list here. I'll put the link to the list on the presentations page. You can also find it at our website, antibodyengineers.org, but I'll put that into the chat later on. Okay, so what did we learn? Well, we learned that the faculty really, really liked it. The industry people really, really liked it. Many of the participants were a bit surprised by how much work was involved and the time commitment involved. In fact, that was one of the biggest challenges because students would sign up. They don't read anything, of course. So when you tell them that this is a 8-hour-day commitment, yeah, they take classes, they have work. They find out that, yeah, maybe it is an 8-hour-day commitment. We found sometimes the team compositions were hard to predict. We would look at the teams and what they wanted to do and put teams together, and then we'd find maybe 3 of them couldn't show up or didn't show up if they were students, and some of the faculty too. So it wasn't, so sometimes we had team leaders that ended up with a team of all high school students, all beginners, which they weren't always happy about. But, you know, we go with it, and we're always flexible. So sometimes great, sometimes not. Our initial hackathon, we had a really interesting surprise. We had a guy join the hackathon and then tell us a week before that he had 2 classes of high school students who were going to be joining the hackathon. So 79 high school students. And this was interesting because we learned that California schools aren't allowed to use Slack, which was one of our major communication tools. So their teachers could, but they couldn't do this. And they also were not allowed, that they also could not be allowed, encouraged to participate or required to participate outside of school hours. So that one turned out to be interesting too. But the thing is, I ended up on the advisory board for this high school, and this was a few years ago now. They still talk about that hackathon, and the students really, the ones who got into it really liked it. They really enjoyed the chance to actually talk with and collaborate with scientists from the NCBI and give their input on software and be treated as professionals. So it was a great experience. It was a bit much for us, but if there are people that want to do hackathons with high school students, and I think some of them ended up going to one of the community college programs, it's something that you might want to consider, and we have some ideas that we could help you with. In general, though, we were trying not to have high school students because we found they were the most likely to flake out and not show up. But, we got some really good ones too. We also ended up having a lot of international applicants. We turned down most of them and made sure our rules specified that we were not taking international applicants. But we ended up taking some anyway, mostly if they were in the same time zone, as we learned time zones could be a challenge. Okay, well, so over the years, we did 4 hackathons starting in 2022, and our last one was 2024. And there's some changes that we made based on what we learned. We learned that our original schedule, we had a lot of talks in our original schedule, and we found we cut a lot of those in the last hackathon because we found the teams really wanted to have time to work together. And having a whole lot of talks, it took that away. We had one person comment that their advice was to have some kind of container so they could use the bathroom and they didn't have to leave the hackathon. So anyway, we learned about that and made sure we put in more breaks, and we didn't have quite so many talks. At our first hackathon, our leaders were not particularly experienced. Todd and I and Sheila Vemu had attended one. Todd had attended 2, I had attended 1, and Sheila had attended 1. So we kind of threw people in there, not really knowing what to expect. But over the years, people got to be more experienced. We added more team leaders. We increased the guidance for our team leaders. Like I said, we had meetings at the end of the day for all of our team leaders. And I think this helped a lot. Our original projects weren't always as well developed. Towards the end, we had a variety of projects at different stages, and some were from previous hackathons. So some went over multiple hackathons and became more developed, and people had a better idea sometimes what they were doing. Our participants weren't always as prepared as we liked, so we added the orientation. This didn't always help with the students because, again, they don't read, they don't always attend orientations. But it helped with some. And we added a Canvas course so that we could have more information ready to go for people. We have had a lot of help over the course of the hackathons. Our team leaders have been Erin Kabajaya, Maggie Bryans, Feather Ives, Sheila Vemu, Todd Smith, co-PIs and team leaders. Alyssa Dillman is a hackathon expert from NCBI who helped us, or from NIH who helped us out, and Kandaya is our evaluator. And then we had a lot of new team leaders come on board. Erica Lannon and Danny Kiner and Thomas Honorato and Melanie Stegman and Eric Olson and Dylan Bolsecco and Lori Kelman and Leah Klein. And Angela Lee was a student who organized meetings on her own, and Jaya Wong from NCBI, and Jacqueline. And I know I'm missing people. Now I'm just going to quickly go through the projects and then Todd can take over. So Erin was working on screening assays for engineered antibodies. Danny was working on making antibodies in spirulina, a blue-green algae. Erica was working on pathogens and whether antibodies to pathogens would work or the pathogens would be able to escape. Sheila was looking at the epitopes that antibodies bind to. And this has become really important lately. There was a paper that just came out with a very exciting, with an RNA vaccine where you make antibodies that triggers antibodies to these neoantigens that are involved with pancreatic cancer. And pancreatic cancer is one of the kinds of cancer that people have really had a hard time successfully treating. So this is pretty exciting. Thomas Honorato and Dylan Bolsecco worked on making antibodies to echinoderms. It's If you have a catalog, you can buy antibodies to mice and humans, but it's really hard to find antibodies to sea cucumbers or starfish or things of that sort. So they were working on could they find existing antibodies and modify those. Melanie Stegman was developing a protein poker game to see if antibodies and antigens would interact. Todd is going to talk in just a minute about whether or not you could take an antibody sequence and figure out what it binds to, or if you could figure out which T cells might kill tumors. And after Todd, Maggie is going to talk about the cells that make antibodies and her projects in working on these cells and their ability to produce antibody and characterizing those cells. Okay, so remember, Innovate Bio Hackathon next spring, this June in June, it'll be a great opportunity to come work with your colleagues and have a good time. And now I'm going to turn it over to Todd and stop sharing.
Nobody said, Todd, you're muted. So antibodies and AI. So Sandy said we don't require coding, but we do have coding projects. So the idea here is, to introduce students, and we have coders that work on these, to data science and machine learning concepts used in antibody engineering. So the rationale for this is you can't pick up a paper or read the newspaper anymore and not hear about how artificial intelligence is impacting everything we do, and in particular in biotechnology. So how do we demystify this? Because I review grants for NIH, or maybe I used to review grants for NIH. We'll find out how that world changes. But we see a lot of machine learning and AI, and these grants come in and nobody knows what, half of them don't know what they're talking about. They don't know how to train a model. They don't know how to evaluate what the model's doing. And so they're, fundamentally weak proposals. And so what goes into these kinds of things? And, and you'll hear ads on NPR about C3PO or whatever, and they say hallucination-free AI. That's not a thing. It's all probabilistic. All of your algorithms will hallucinate at some point. But in antibodies, there's really two grails, right? So one is this concept of a universal diagnostic. That is, given an antibody or a T cell receptor sequence, can we predict what antigen it binds to? That is, from a drop of blood, we could give you a profile of your disease history or even active diseases based on those antibody sequences that are in that drop of blood. Universal design is given an epitope. Sandy showed, the epitope, a little bit of the protein that the antibody binds to. If we have an epitope, can we design an antibody de novo? And so that is, could we put this sequence into a program and then get out candidates for antibodies? And this is an area of very active research, because you can imagine with databases like the epitope database that Sandy showed, there's a lot of things that are understood. And if we can make these antibodies more quickly, we could design new kinds of therapeutics. So the state of the art today is that universal diagnostic.
4711. Okay. All right. Bye.
Somebody should mute Linnea. So we call it immune profiling. So that
Sorry.
I get a collection of, I get blood cells, I isolate the RNA, and I do a bunch of sequencing in mass, at scale, massively parallel formats. And what I get is I get a data set that tells me about all the antibody sequences in there, or T cells. And from those, I can do things like, I don't want to annotate. Never mind. Just looking for my laser pointer. Okay, but anyways, in there I can get biomarkers out, but I also get all kinds of sequences that I can use for machine learning for that universal diagnostic. On the machine learning front, people often design antibodies in mice. And then what they want to do is they want to turn them into human forms so they can use them in therapeutics. And so we call that humanizing. We also can take sequences and then improve their properties, either binding or developability or specificity. So lots of things going on there. And then this de novo design in COVID-19 and flu, where we've had a lot of antibody sequences, we've seen some progress. And then, as Sandy mentioned, these hackathons can unfold over multiple years. And so this is just showing a page from the GitHub. We started in 2022 with this project, COVID, Not COVID. That led to 2023 immune profiling, because I learned that we weren't really quite ready for machine learning. We needed to understand the data first that feeds into these algorithms. So we did that, and then I did some other work with NCBI with a hackathon, and we finally got to Antibodies and AI. I want to point out Jetstream, the National Science Foundation, has free computer resources that you can apply for and use in these kinds of projects. And so we actually had some very powerful computers to do this work on that had both GPUs or CPUs, but we're talking on the order of 64 cores, 256 gig of RAM, a lot of kind of macho IT terms there, but they're very powerful, and we can use them for these projects, and simple application process. So this year, what we did is we ended up with two parts. One was a colleague of mine from my Geospiza days. He was the one who actually developed the Gene Sifter gene expression analysis platform that was used throughout research and industry. And so he's a gene expression expert. We adapted that to next generation sequencing in 2010 or so. And he's continued in companies doing this kind of work, and got interested in the single cell sequencing problem. And the idea is, can we identify biomarkers with active T cells? So if you sequence DNA from individual cells, you can actually put the alpha beta chains together, or the heavy light chain, and that gives you a lot more information. You can also measure the population of those cells more directly than inferring it from the sequence data. So that's pretty cool. And then on the machine learning, what we wanted to do is can we generate an antibody sequence to a COVID spike protein? So I was leading the machine learning, Eric was leading the immune profiling, and our team had these individuals, and you'll have all the slides available. You can look those up later. And so, as I mentioned, with the immune profiling, is what you want to do is look at these best TCR clones. And these are going to be ones that are going to be rapidly expanding in the case of cancer. So if you have data sets from people that don't have cancer and a particular kind of cancer, you can then look at the RNA sequence data, single cell RNA-seq or scRNA-seq, and make some predictions about which clones are the ones that are responsible for the T cell response to that cancer. Those would be the receptors that you'd be interested in, either following as biomarkers or using those sequences to maybe develop something therapeutic. And so what it begins with is these tables. You have large tables of data, pre and post, treatment kinds of things, the clone counts. And so this is looking at anti-PD-1. So we're looking at the T cell response to one of these cancer inhibitor drugs where you're stimulating the immune system. And basically you have all of these sequences, you have information, some metadata about those sequences. And what you want to do is feed these into a program. We were using ScanPy, which is a single cell analysis in Python. Eric is an R expert, so he was very curious about how these Python tools are stacking up to R tools. And so that was really a lot of the nature of the project. And what you do is you generate these kinds of output where you're looking at, you can't see my mouse, but you're looking at, principal component analyses, asking questions about how these sequences cluster in space to tell you which things are related to one another and which ones belong to similar kinds of clones. And then you can look at the gene expression of those to ask the question about, which TCR sequences are expanding in those cells. And so you can either use gene expression of various genes in those cells, or the TCR clones to identify biomarkers for particular cells. And if you could measure these biomarkers, then you could say, oh, is this patient actually responding to the therapy in a positive kind of way? And so I'm not going to go through all the analyses, but this is the kind of output people deal with. When we go on to the classification of the antibodies, we had two parts. One is, given a data set of COVID antibodies, can I predict neutralizing from non-neutralizing kinds of sequences? And then can we use the spike protein and generate a sequence? And so this is a flow chart that we go through. We have a data set that goes in. We can ask some questions. We have it annotated in terms of neutralizing and non-neutralizing. We can split it into two parts. So one goes through training. We use a program called AbLang for embedding. So this is kind of where we get into the nuance of machine learning. How do these models work and how do you train them? We don't, in bioinformatics, we compare sequences to one another. We say, does an A match an A? Does a C match a C? You don't do that in machine learning. What you need to do is convert these into numerical values, vectors. And these vectors can be large dimensional data sets of themselves, like 480 attributes, and then numbers for each. And you have to make sure your data are well separated so that things don't overlap. And then you do your machine learning on those data sets. And so these embeddings are where some of this logic is put in place in terms of the biology and how that works. And so we go through, what does that mean? And then the second part was generating. So I mentioned, this concept of embeddings. We use data sets from this wonderful resource. It's called OPIG, for the Oxford Protein Informatics Group. And they have all these things that, COVABDAB, everything goes ABDAB, so that you can all babble together about antibody databases. And then this is one of the notebooks from some previous work I had done with NCBI and professional software engineers explaining these concepts, right? And so all of these notebooks are available in our GitHub repository. Again, we go through, there's a lot of different things you can do to play with the data and learn about different kinds of methods that are used in machine learning, accuracy, F1 scores. These are all parameters that tell you how well your model's working. You're looking for scores that are above 0.7 kind of thing. And then these NN shallow 2, and so forth. These were all experiments that were done by one of our participants in terms of taking that data set, the COVID data set, and just looking at different attributes of the data and organizing it, and then rerunning the training to see which ones actually had some meaning. And so those are the kinds of things that we can do, that get students involved, is you take a data set and, sometimes these tables can be millions of rows and hundreds of columns. So what columns actually do you use to get meaningful information from the data to train a model? And then finally, one of our colleagues, it always helps to have a postdoc from NIH on your hackathon team. So he said, I want to do the machine learning. And he used RF diffusion, which is, it's a cool algorithm. Basically, it came out of image analysis. And what you do is you make your image real blurry, and you make it blurrier and blurrier and blurrier, and you train a model to see if it can deconvolute that blurry image. And that's actually how things like DALL-E work. When you're asking, ChatGPT to make an image, it had this database of these images that it blurred and then trained models. And using that, it could generate then an image to your prompt. In our case, our prompt is an epitope sequence. And what we're showing here is this cyan. The lighter green is the native COVID antibody. The darker green is what was generated by the algorithm. And one of the things that has come out, and this is February 2025. So just to let everyone know, we are on the cutting edge, right? So RF diffusion by the Baker Lab, so here in Seattle, Protein Design Institute, they're designing antibody sequences and using yeast display. one of the things I like to point out to people is we don't get an answer from AI, we get many answers. And what we can do today is we can use high throughput methods to then interrogate, to test the hypotheses that are generated by the computer. And so that's exactly what they did with yeast display. And then they looked at hemagglutinin difficile, toxin B, respiratory syncytial virus, and then of course SARS-CoV-2. And they were able to identify antibodies that would be neutralizing in all of these particular cases. So 2 outgroups, if you will, that aren't traditionally studied, they were able to do with this method. And so we used actually some of their previous RF diffusion code in the test that we did in our hackathon. So basically, to summarize, there's lots of things we learned. One of the things I do on these projects is I'm the team system administrator. So I set up and provision the computers and get everything, and I have all the instructions in those GitHub notebooks, and I keep people working is kind of my job. And you can see there's a lot of things that we learned from this, some things that we'll save for future discussions in technical nuance. So anyways, that's what we've been doing on machine learning and antibodies. And with that, I will stop my share and Maggie can jump in.
Thanks, Todd.
Okay, this is way too cool. I want you to do more hackathons.
Well, thank you for bringing that up, Linnea. We are in the process of getting a supplement together, and hopefully, maybe we'll have one this summer. Hopefully, and if not this summer, we are doing an Innovate Bio one for sure next spring. So
I want to
Go
be
ahead,
part
Maggie.
of one before June. It's so fantastic. Machine learning, you're covering so many bases with what you're doing. Hey, I still can get excited even if I'm old.
That's the goal.
Yeah,
Okay,
and-
Maggie, go ahead.
I'll chime in and saying it is a really good learning opportunity for any participant, because you get to learn what every other team is doing with all of the report outs. So I learned a lot from Todd and what his team was doing, and from others that are using really neat software to look at, antibody epitope interactions. And I actually introduced that into my research class. So it is a learning experience for all. I'm going to talk about NISTCHO cells and CNIS-MAB. And this was the work of 2 hackathon teams, and Sandra called these teams the Dream Team, which was a great group of people that came together. Again, we come together for 4 days and have a great time developing curriculum. And in this case, we were developing wet lab curriculum that could be used, incorporated into courses, or used, as Sandy mentioned, as an independent study. And that's typically what I've used these projects for. We have an internship course. It's a 6-credit course where students can do an internship project independently in the lab. So these hackathons have been a great source of ideas for these independent study type research projects. So we had a wonderful resource to work with and have fun with developing these projects, and that was the NISTCHO cell line. So NIST, the National Institute of Standards and Technology, along with Millipore Sigma, developed this standard cell line, and they made it available as a research grade test material. And as such, all of us could get it for free by signing a contract saying we would provide some information about the cells as we grew the cells and froze the cells and so on. It's a wonderful resource for teaching biomanufacturing. And, late in 2023, Laurie Kelman and I had a NIMBLE grant that funded us to to do some of these research projects, but also to develop directive documents to use these cells in biomanufacturing and biotechnology training and education. And we posted those resources, and they're available at biomanufacturing.org. So this cell line is wonderful. It's a modern CHO cell line, finally, we can all get our hands on and use for our biomanufacturing and biotechnology courses. It produces a monoclonal antibody, again, highly relevant, called CNIS-MAB, which recognizes glycoprotein F on the outside of RSV. So it almost became a product. It got to the original antibody, got to sort of a phase 3 clinical trials by a company in the past. And that cell line was then donated to NIST. And they then developed the NISTCHO cell line from some of that technology. So we were all able to get our hands on this and work with it. So over the course of those 2 hackathons, we came up with many, many ideas for wet lab research projects, and these are some of them, but not all of them. The 2 highlighted in red are 2 studies that we actually took to completion in my labs, and the students were able to publish that, as Sandy mentioned, in the JATE journal. So I want to talk about them. And then in orange are 3 projects that really, some were started in the first hackathon, but really fleshed out more on the second hackathon. And that was developing a functional assay for CNIS-MAB, developing a titer assay that we're all looking forward to getting to, and also to look at the effect of glutamine on NISTCHO doubling time, and I'll explain why glutamine in just a moment. We also had great ideas for introducing some molecular biology type experiments with NISTCHO cells, some genetic analysis of the insertion site, and looking at the cassette that was introduced, and so on. So we had a great time coming up with all of these potential projects. So I'm going to talk about the stability studies first that we did, and then go through some of the others. So biomanufacturers are required to perform stability studies on the production cell lines. So in an upstream process, depending on the scale, they might be growing these cells for about a month in culture. In some cases, that could take the cells up to 60 generations, 60 doubling times. And tests have to be done to ensure that the monoclonal antibody is still being produced. You know, the cells didn't lose the transgene, and they're still producing the MAB at high levels. So we designed a project to analyze MAB production in these CHO cells up to 100 generations. We actually went slightly over that. So this project being long-term, I took it over 2 semesters. So we had 1 student working on it in the fall semester, and a second student picked it up in the spring semester as they both did their internship projects. And this is the design of the experiment that we designed during the hackathon, where we would take a low passage number NISTCHO cells from our cell bank, resuscitate the cells, grow them long-term in culture. So they would have to grow them for the entire semester in culture. And every week or so, every 6 to 7 doublings or generations, they would freeze an aliquot of cells. So they had cells that were then frozen down at different passage numbers or generation numbers across the whole length of the project. So they would freeze the cells, and these are cryovials in the middle here. And then once they had set up all had representative cells that were frozen from different passages, they would then carry out a normal cell culture of those cells and monitor the behavior of the cells, you know, monitoring how the cell viability was, the maximum viable cell density, and importantly, if they could still produce monoclonal antibody, which is really what we were testing. So that was our design of the experiment, and this is some of the results that were published. And there's a lot going on in this graph in particular, but these are typical cell culture growth curves where you have lag, log, plateau, and decline phase. And the different colors of the growth curve represent cells of different passage number. Imagine these are young cells, these are older cells. These have been in culture for 20 some weeks. This is our initial culture, passage 10. So the P stands for passage number, and we can relate that back to cell doubling. So overall, when you look at the comparison of the growth curves, you can see they're very similar. There's not much difference between the dark green initial cell line, and the sort of a purple color, which is the cells that have been that are at passage 46, been in culture for a long time. So the performance, even although they've been in culture for an extended period of time, is still very high. Similarly with their cell viability, again, the different cells from different passage numbers are growing almost identically throughout the culture until they run out of nutrients and they drop off. Importantly, we wanted to look at monoclonal antibody production. So during, we collected the medium that contains the monoclonal antibody, and we quantified the MAB in each of the cultures. And across the bottom here, these are the representative number of cell doublings that corresponds to the different passage numbers and the level of monoclonal antibody being produced. So these cells, this tells us that these cells are very stable. They maintain possession of the transgene, and they continue to produce monoclonal antibody even after over 120 doublings in culture. So this was important information. None of this had been published for NISTCHO cells. The good thing about the cell line was it was very new. There was no publications out there. So it was that great feeling for the students to be able to be producing new information that would be of interest to others. So that summarizes the stability study that we designed and then executed. The second study we came up with in the first hackathon was to do a cryopreservation study. Again, this information wasn't known for NISTCHO. We didn't really know how they would behave after being cryopreserved at different temperatures. So we decided to do a comparison study. And this really came from the fact that many community colleges who were adopting the cell line cryopreserve their cells in the -80 freezer. That's what they have for freezing. And as many of us know that have grown mammalian cells, this is suboptimal for some mammalian cell lines that just don't do well unless they're in the vapor phase of a liquid nitrogen tank, which is about -196. We have a -150 freezer, so we have close to the ideal. So we did a set of experiments to compare this cell behavior after they were cryopreserved at -80 or -150. And so our approach was to create cell banks for NISTCHO cells at the two cryopreservation temperatures, and then at 2-week intervals, the students would thaw a vial of cells and culture them for 9 days, going through the entire growth curve, and then compare their viability, maximum viable cell density, and of course monoclonal antibody production. So that's the design of the experiment we came up with, and these are some of the results from that study. So what we're seeing in the top two graphs is a comparison of the two cryopreservation conditions. This set of growth curves from cells preserved at -80, this set of growth curves from cells preserved at -150. And you can see just by looking at both charts that they're very comparable. You have the normal variation from lot to lot, if you like, for the cryopreserved cells. And what we're seeing with the different colors of the growth curves are the length of time they've been cryopreserved. So these cells have been cryopreserved for 2 weeks, this kind of dark blue color. So you expect them to be behaving as well as can be. And then going all the way up to week 30, again, this is over approximately 6 months we did this study. So again, a 2-semester study for these students doing their internship project. And we can see, comparing both cryopreservation conditions, they're very similar, which is quite reassuring that you can store our cells at -80 for up to 30 weeks at least anyway. And then we also wanted to see other parameters, the initial cell viability with some variations at the beginning where the students were really perfecting their techniques. When you look out to 30 weeks, with orange being -80 and blue being -50, they're very comparable, not much difference at all. Similarly, with the amount of monoclonal antibody the cells can produce, with again -80 being orange, -150 being blue. As we go out towards 30 weeks, we see some difference between the amount of monoclonal antibody produced, but that could just be, variation between the cultures and not anything really to do with the cryopreservation, because you can see otherwise the cells are performing quite well. So the students, again, were able to publish this in the JATE journal, and again, bring new information to the community as far as adopting NISTCHO cells and how we can cryopreserve them. So that was a cryopreservation study. Moving on to our second hackathon, many of us were looking to develop a titer method for NISTCHO and to have that type of SOP that we could share with others. The exciting thing about this experiment was that we actually had real-time experiments going on. So we had Leah Klein and her technician Sandra from the Waters Corporation as part of this hackathon team. So as we discussed conditions and optimized parameters for this analytical assay, they were actually doing the work real time. So that was quite exciting. So a titer assay then is an assay that's going to allow us to follow and determine the concentration of the monoclonal antibody in the cell culture medium. So when the CHO cell produces the MAB, it secretes it into the medium, making it much easier to collect and purify in downstream processing. So typically companies will follow the titer, the amount of antibody that's being produced as the cells are growing in culture. We don't have one of those assays for NISTCHO cells, and we really need one for our teaching and our courses. So on the team, we had Donna Barron, who has an analytical chemistry background. So it was great to have her expertise in HPLC as well. So the way it's typically done and the way it's done in industry, speaking to our SMEs that joined in for some of the days, is through a protein A column, an analytical protein A column run in an HPLC instrument. So we were developing the initial parameters for that, researching the literature, coming up with a good set of starting parameters that we would then work on to optimize as the days progressed in the hackathon. So Sandra and Leah were presenting their data. So this is the peak of the CNS MAB coming off the column, and we would discuss their data as we went through. So that's something that both Donna Barron and myself are working on to optimize this and to create an SOP that we can share with others for their sort of analytics as they go through their biomanufacturing course. Another need for the community was to have an ELISA assay, a functional assay for CNS MAB. And this is something that Roger Greenwell worked a lot on in the hackathons. We were able to do research and find a target peptide or a peptide that could be used as the epitope for the CNS MAB. And we synthesized the peptide, and we're working on that now, developing that assay. So the way that would work would be our peptide that would be recognized by CNS MAB will be bound to the 96-well plate, and then we would use our media from the CNS MAB culture as our primary antibody, or from the NISTCHO culture as our primary antibody, and then again as a typical ELISA binding secondary antibody with horseradish peroxidase or some fluorescent tag that we could quantify the amount of reaction that's going on in the 96-well plate. Roger put in a lot of time developing some molecular biology, and he's actually using that in one of his courses to actually express the peptide that would be his tagged and purify the peptide, so we could actually then disseminate that as a resource to other community colleges that didn't have the resources to purchase peptide, which can be quite expensive. So that was another one. So Sandy, I'll pause there.
Yeah. Okay. I know you guys did so much.
it was really a lot of fun and quite useful for the community. So I'll stop sharing and go back. Let's go back to Sandy.
I don't have anything. I mean, I have all kinds of things to add, but not time. Not time to add them all. So let's take a couple questions. No questions now? I know Lori was asking about the June hackathon that we're planning with Innovate Bio. That's going to be 3 days, not 4. It would be 8 hours a day. So the same kind of time commitment. We'll start, we're going to be talking about this at High Tech and recruiting team leaders. I'm sure Maggie will be one, right? Maggie will be one. And yeah, so if you have ideas for things you'd like to work on with your colleagues, let me know, and we are happy to. We'll set up a team. So
I think it'd be really interesting to some of your topics are things that need to be explored more for curriculum for programs like machine learning and AI. And then, I mean, essentially what you're doing, a lot of things that you're doing could be curricular topics that need to be expanded on and shared with the community. They're like all separate little grants.
I was
Totally.
waiting for you to say that, Linnea.
I knew.
Yeah, I mean, and so yeah, I mean, it takes one of our community college partners to step up and say they want to do it, and then I think there's teams that can help them get there.
In fact, one of our partners did apply for an ATE grant. We haven't heard.
Yeah.
But at least one ATE proposal came out of this.
Yeah, I think more could come out of it. And then maybe there needs to be, if you're not a supplement, it might be good for, and I can talk to Celeste about that, because I could see where you could have a face-to-face meeting with quite a few people to work on the idea of developing maybe a supplement off the center.
Yeah,
yeah,
possibility. I should mention too quick, some of the people here who are here today, Daniel Moore and Christy Shuda-McGuire and Dylan Bulsacco and Hital Doshi, and I think Nick Reeves were also hackathon participants. So it's great to see you guys.
Well, I'll gladly help with this, because I just think you just hit the nail on the head with like several times.
Well, what I've learned, what we've been doing is each hackathon, we look at our feedback and we try to tweak the next one. What we're hoping to experiment with more with the next one are some more community building activities between people who are participating. So to have a speed networking session among hackathon participants, so they get to talk to people from other teams a little bit more. And then, we've been doing a lot to try to get our team leaders to have a chance to talk to each other.
It's like using hackathons to develop emerging technology curricula.
And faculty research professional development methods too, right?
Yep.
It's not just
no.
a good workshop.
I know.
Beth.
I think one of the really fascinating things besides the science, obviously, is when you started out, Sandy, talking about all the different roles that people play, that it's not just sitting there at a computer typing, which would be, I think, quite a draw for the folks who are looking at it going, well, I can't do that. I have nothing to do with that. So in some way to amplify that aspect as well, because it's just a microcosm of, biotech in general. So.
Oh yeah. And it helps. It really gives us a chance to integrate people with different levels and kinds of experiences. Like each team had people from industry, sometimes our subject matter experts, sometimes, our faculty and students, and everybody had something they could do and be productive and contribute because they had different roles.
Yeah, Sandy and Todd put together a really good advisory board for this project. So they were already invested, so we could pull on them. We could actually have them join the hackathon team, and somebody with 30 years experience in map production and industry was a great resource to have as we develop some of these projects. Also, Leah Klein, who was doing the HPLC, is part of the advisory board. So it was a great combination.
Well, yeah, we were lucky in that regard. But we spent a lot of time recruiting.
Sandy, Laurie Lindbergh has a question in the chat for you. Will you send us general outreach to the hackathon?
Yeah, totally. Absolutely. I'll also post links to all the resources that have been developed when I post the presentation, and make sure we put that information in the newsletter and on the website as well. Oh, Linnea, you look like you were saying something. But you're muted.
So I'm sitting in a car. Anyway, my life lately. This could be, yes, it's under biomanufacturing, but actually career and entrepreneurship, your hub, this is definitely hub material to be expanded.
Well, see, I get people asking me, one of the things I always tell people in the community college courses is that get to know your classmates, because there's going to be a time when you're going to be calling on them to find your next job, right? It's going to be really important to develop your network and show each other that you can work together and help each other. But I get sometimes people coming to talks that they don't have that opportunity because they've already graduated, or maybe, it's past. Okay. So I said, be at a hackathon, join a hackathon, meet people.
So another paper, the new professional development for faculty.
Yeah, how to work in teams. It's not something it's funny because we're all accustomed to being leaders, but we're not accustomed to being team members. And it's not as easy as you might think.
So Sandy, Todd, and Maggie, thank you for such a great presentation this afternoon. And Maggie, I'm sorry that you had to cut your part a little bit short. I wish we had more time. But I thank you all on behalf of the entire Innovate Bio leadership team. Thank you for joining us this afternoon. And we wish you a peaceful weekend, and we'll see you next week.
Thanks, Dion.
Have a great weekend, everybody.